Welcome to Lecture 7, where we will go over the diversity and characteristics of fungi. Fungi are considered a highly diverse kingdom and are found in nearly all aquatic and terrestrial habitats. They can be unicellular or multicellular. In addition, many of them play important roles in the ecosystem as decomposers, where they break down organic material and help release it to recycle nutrients back into the ecosystem. Fungi are considered to be heterotrophs and absorb nutrients from their environment by secreting hydrolytic enzymes to digest various types of compounds. As stated earlier, many of them are decomposers, but fungi can also be parasites as well as mutualists. Fungi typically exist in single cell or yeast form. This is their typical form when they are in moist environments when nutrients are readily available. However, if nutrients become less available, they may form multicellular filamentous hyphae to aid in the acquisition of nutrients. These hyphae are considered to be tubular cell walls that are made of chitin. This surrounds the cells to provide strength and aid in feeding. These hyphae can be septate, which means that the individual cells are divided by septa, or suinocytic, which means that they are joined multinucleate cells that form the hyphae. As nutrients accumulate in the interior portion of the cells, water also moves into the cells, creating osmotic pressure, similar to what we see in plant cells with the plant cell wall. Hyphae can also form a filamentous network. In that instance, we would call the network a mycelium. This serves to increase the surface area for feeding, once again aiding in the acquisition of nutrients. These hyphae can have rapid growth as proteins are directed towards the tips of the hyphae. As a result, the fungi invest energy into extending these networks rather than growing larger hyphae. This can create a very extensive network below the surface, as seen in the diagram to the right. In addition, these hyphae may be highly specialized to carry out different functions. Some hyphae can be adapted for trapping and killing prey, while mycorrhizal fungi have specialized arbuscules. These are hyphae that are adapted to exchange nutrients with plant cells, since mycorrhizal fungi have a close association with plant roots. This is an example of a relationship called mutualism. Hostoria, which are similar to arbuscules, are specialized hyphae that can extract nutrients from plants. Mycorrhizal fungi are found in close association with many vascular plant species. The plant acts as a host and receives the benefit of having an increase in the absorption of vital nutrients such as minerals and ions due to the presence of the fungi. The fungi, on the other hand, receives a rich supply of organic compounds. These fungi typically recolonize the surrounding soil through spore dispersal. There are two main types of mycorrhizal fungi. Ectomycorrhizal. These form sheaths of hyphae that cover the roots and penetrate the root cortex. However, they do not enter into the plant cells. Arbuscular fungi, on the other hand, extend their arbuscles to penetrate through the cell wall of plant root cells. Fungi can reproduce sexually and asexually. The diagram below represents a typical life cycle of the fungi. Note that the haploid state is in blue, the diploid state is in pink, and the heterokaryotic state is in purple. The mycelium, which is a network of hyphae, can produce spore-producing structures in asexual reproduction. These will then produce spores, which will germinate in the surrounding soil. The mycelium can also undergo fusion of various types of cells to produce the heterokaryotic stage. The nuclei may fuse. This is a process called karyogamy to produce a zygote, which is diploid. This zygote can undergo meiosis to also produce spores, which are haploid. These spores can also germinate to produce more mycelium. During reproduction, hyphae can release pheromones. If the hyphae are from two different mating types, they can fuse using binding proteins on their cell membrane. However, if they are from the same or genetically similar mycelia, fusion will be prevented. This is a mechanism to ensure genetic diversity. In sexual reproduction, the hyphae can release pheromones to induce the sexual phase. This will cause the fusion of the cytoplasm or plasmogamy between different mating types. This would then form the heterokaryon, which has two separate nuclei. We term this heterokaryon dikaryotic, meaning two nuclei. Over time, if the nuclei fuse through karyogamy, this would produce a true diploid cell, or the zygote. Once again, this zygote would then undergo meiosis to form haploid spores. Note that these spores would be considered genetically diverse. These spores can then colonize new areas and undergo germination to form new mycelium. In asexual reproduction, the deuteromycetes refers to any fungi that lacks the capacity for sexual reproduction. This means that they would not be able to undergo karyogamy or form heterokaryotes. Molds are considered to be filamentous fungi that can form visible mycelia called conidia. These conidia produce haploid spores through the process of mitosis. Yeasts can also undergo mitosis to form buds from a parent cell, as seen in the diagram to the right. Some fungi can actually switch to asexual reproduction if a genetically different organism is encountered. However, for the most part, they would reproduce asexually. If you remember from the previous lectures, fungi are considered to be members of the Opisthokonts clade. This means that they evolved from a unicellular flagellated ancestor. The oldest terrestrial fungi fossils are present in the fossil record approximately 440 million years ago. It is believed that the ancestor to fungi was likely an aquatic single-celled flagellated protist. This means that fungi are actually closer to animals than they are to plants. This is also supported by DNA evidence, which suggests that fungi are closely related to nuclearids, which are unicellular protists, while animals are closer related to choanoflagellates. This finding suggests that multicellularity actually arose separately in evolution. Fungi are considered to be among the land's earliest colonizers. It is estimated that land was originally colonized approximately 505 million years ago. Soil samples during that time have a signature chemical profile which is very similar to the environment that we see around active living fungi. It is believed that land was colonized by a green slime. This green slime consisted of many different organisms such as cyanobacteria, algae, and eutrophic species such as fungi. Early fossil evidence suggests that the first ancestral fungi formed a mutualistic relationship with early land plants. Plants originally invaded land approximately 470 million years ago. If we take a look at some of the early fossils of plants, such as Aglaophyton, they have hyphae, which is shown to have penetrated plant cell walls. This is very similar to present-day arbuscles. Genetic analysis also indicates that SIM genes, the genes that are responsible for mycorrhizal formation, were also present in the common ancestor to land plants. This strongly gives evidence for the presence of mycorrhizal fungi. Also note that many early plants lacked a root system. Thus, having a close association with mycorrhizal fungi would have been a very important way to acquire nutrients. Over the past few years, metagenomics has completely revolutionized the way that we classify fungi. Many traditional groups, such as the Zygomycota, have been abandoned and their members are assigned to other lineages. Some lesser-known groups, such as microsporidians, may actually represent the basal fungal lineages. There are currently 145,000 known species of fungi. However, it is estimated that there is close to 2 to 3 million species. There are also approximately 2,000 new species of fungi discovered each year. This diagram represents the current phylogenetic hypotheses representing the relationship between the major clades of extant or living fungal groups. Note that we have an common ancestral protist, which was considered to be flagellated. This then led to the two basal lineages, the cryptomycetes and microsporidians. We then have the chirtids, zupagomycetes, mucoromycetes, ascomycetes, and basidiomycetes.